Hi, so in this video, we're going to talk about the dihybrid crosses and how that's different than monohybrid crosses and two Mendelian laws. So going back to your monohybrid cross, if you watched the previous videos, if we were just to look at this as an example, this is your just any somatic cell that's found in your, I guess, testicles or your ovaries that's going to form sperm and egg. And we talked about meiosis. So inside of those somatic cells, remember that it's a diploid cell. So you have both sets of the chromosomes from the parents. So then how or what happens after with these chromosomes? And you know that these chromosomes, if you were to randomly select them, these two can be, this sperm can have the A allele, randomly this one could have the this type of allele, this one can have this allele, this one can have this allele, and then this one can, this is just all based on random, right? So each of these sperm is going to have one set of these two alleles, making each of these sperm diploid. So that is actually a law, or called the law of segregation. That means that the law of segregation, it really states that the allele pairs separate randomly during meiosis. So if you look at this is your allele pair. You can have this type, this version of the allele, and they actually will segregate to go individually into each of the sperm. So that sort of leads up into your Punnett squares, because out of this pair you can have either the capital A or the lowercase a. But then how is that different than if we were to have two sets of allele pairs? So if you were to have this, the regular set from the first slide, and then now you added a new one. So what would happen if meiosis were to occur and if this were to split? So then you would have two alleles, but which ones would actually happen? So then if we were to just randomly choose this one, maybe this sperm would have this allele, this one have this allele, this one would have this one, or could have this one, and then I'm just choosing random ones. It could have this one, and then you can see that you can have this one. Let's just choose randomly and choose all these ones and finish all of the sperm cells that we have here. And you can see that the question is, well, what's the problem here? Is this accurate about how these allele pairs actually got divided in meiosis? And you can see that this actually has an error, because you can see that this sperm cell right here has two of the same allele, like both sets of the same allele pair. You have your uppercase and lowercase. That means that if this genotype were to be accounting for hair color, like brown hair and blonde hair, what would happen? You actually have both sets. Or this one, same thing. If you have blue eyes or brown eyes, well, then you have both sets for eye color in the same sperm. How would that happen, right? So this actually doesn't work. So what Mendel actually found out is that the second law is called the Law of Independent Assortment. So what independent assortment is, that these allele pairs will actually separate independently from each other or from other allele pairs. That means that if your eye color, you have traits for eye color, that will separate independently from hair color. So you would not have things like two of the same ones, while you should have one of each different kinds of allele pairs at a random sort of same equal probability, same chance. So this is your example. So what we would do correctly is that you're actually going to select the same one from each and looking at all the combinations or possibilities. That means that if I were to look at this independent from this one, well, I can either have a uppercase A from this set and a lowercase t from the other set, or uppercase A from the first set or uppercase t from the other set. And then, well, we haven't used the lowercase a yet, so lowercase could also happen, and then lowercase t could also happen as that one combination. And then lastly, lowercase a could also be uppercase t. Well, we don't have to fill in these other sperm cells because I have covered all the possibilities of my allele pairs based on the law of independent assortment. So what that means is that the allele pairs will separate independently. Again, this is a allele pair. This is also a allele pair, and all these individual ones will separate from each other, independently from each other. That means that this one and this one will not be found in the same sperm cell, or this one will be, and this one will be found in the same sperm cell, because they have to be separate independently. So that's your second law, and this is actually tying into the dihybrid cross, dihybrid means that you have two different allele sets, whereas the previous one only had one. So if you think about the previous slide, this one only had one set. That's a monohybrid. And then once we get into your dihybrid, you actually have two of them. So right here you have two sets. So how does this lead into a question? So let's look at this one as a question. This one, determine the genotypic and phenotypic ratio between two heterozygous pea plants that are both green and wrinkled. So you notice that there are two different kinds of phenotypes. You have your wrinkled, green, and both of them are two sort of different allele pairs, because this one is for color and the other one is for texture. So these are two different things. And you know that both of these ones are heterozygous. So what that means that if we were to say that green is dominant, this is dominant because we know that yellow, recessive phenotypes are yellow and smooth. So green is dominant and yellow is recessive. And we need a letter for this one. So let's just use the uppercase G for dominant and lowercase g for recessive. And then for texture, we have wrinkled. And this one, we know that it is dominant, so I'll just put a D here. So then this is going to be, let's not confuse with my letters, dominant. So we'll use a W. And then for smooth, this is my recessive. And then we'll use the lowercase w. I'll underline that. So you know that this is going to be two heterozygous. So we know that the first parent is going to be uppercase G, lowercase g, and uppercase W, lowercase w. This is going to be crossed with same genotype, two heterozygous. So how do we set up this Punnett square? And you know that because now we have actually two different letters, two sets of letters, this is going to make our Punnett square a little bit bigger, more boxes. So let's look at this one. So if this were to be the male and this one's the female, let's look at all the possibilities coming from this first set. If you look at from the previous one, you can sort of see that first I need the uppercase G, so uppercase G, and then one from the W, which is uppercase W. That's my first combination. The second one is, this is going to get a little bit messy, but I need a uppercase G, and then now a lowercase w. I'm going to just change color so it's easier to see. Let's use green. And then now let's go to the lowercase one. So we have the lowercase g, but now the uppercase w. So this is uppercase w. And then finally, you have same lowercase g, but then now lowercase w. So this is my heterozygous. So you notice I'm using one from each of these pairs because they are independently assorted. So then this is going to be crossed with my female, and it's going to have the exact same genotype because it has the same genotype. So I'm just going to mimic this one. So there's my GW, and then uppercase, lowercase, and then lowercase, uppercase. And then let's see if I can scroll down here. And then I have my lowercase g and lowercase g. So now I need to fill in this box. So let me change the color again, and let's try this. Here's my grid for my Punnett square, and now I'm just looking at all the possibilities of my genotype. And this is exactly the same as what you did for monohybrid cross, except that you're going to have a little bit more letters. So the first box is going to be this one going in here, and this one's going in there. So I have two uppercase G's and two uppercase W's. So go down the row, two uppercase G, one uppercase, one lowercase W, one uppercase, one lowercase, one uppercase, one uppercase, one uppercase G, lowercase g, uppercase W. So make sure you're underlining your lowercase because it's really easy to make a mistake if you have forgotten. So same thing, uppercase, lowercase. So this is going from this one and this one. Uppercase, uppercase, lowercase, lowercase. Uppercase, lowercase, uppercase, lowercase. Make sure you underline it. So uppercase, lowercase, lowercase, lowercase. So that row is done. And then this is my uppercase, lowercase, upper, 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 lower, upper, lower. Make sure I underline them. And then lowercase, lowercase, and then upper and upper, lowercase, lowercase, upper, and lowercase. And we're almost done. Last row. You can see now we have a lot more combinations now. So uppercase, lowercase, upper, lower. So make sure I underline these. And then uppercase, lowercase, lowercase, lowercase. And then lowercase, lowercase. And then uppercase, lowercase. And then lowercase, 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 lowercase. So this is finally done. So then now I'm looking at the phenotypic and genotypic ratio. So let's look at my phenotypes. So then remember, I have dominant G. Dominant G represents a green one. So let me just kind of scroll back up here again. And then a dominant W is my wrinkled one. So which ones are my dominant ones? So let's look at that. So I'm just going to circle all the ones that are dominant green and dominant wrinkled. So this is a dominant one. So here's one. This is one again. So these are my phenotypes. Here's another one. Right, they're all uppercase, dominant, dominant. This is my dominant. And then this is also my dominant. This one is also my dominant right here. Oh, right here. And then, oh, my pen's not showing up. I don't know why my pen is not showing up. But those ones are my dominant. It's just a little laggy. There's my dominant. Here's another dominant. And I think that's it. So this one is not, because that's recessive, recessive, recessive. And this one is recessive for G's. And then this is my, oh, I forgot to underline this one. So that should be recessive as well. So my phenotype here is, now I need to count the boxes, but these are my dominant phenotypes. That means that I'm going to have green and wrinkled. And how many do I have here? I have 1, 2, 3, 4, 5, 6, 7, 8, 9. So I have 9 of them that are green and wrinkled. What about if I have green? Now I'm going to look at the dominant for color, but now smooth, recessive. So I'm looking at recessive for W. So I'm going to change the color again. And let's see, use the blue. Dominant G, so uppercase G, but then lowercase w. This one is my dominant G with lowercase w. Uppercase G with recessive W, sorry, I should say that. This is not, these are all recessive G's, dominant G's, recessive W's. So you can see that I have 3 of them, so that's 3. Now I need to switch to the yellow one, which is recessive. So this is going to be yellow, recessive for color, and wrinkled, which is dominant for texture. So then how many do I have? So let me switch colors. And you're going to slowly catch a pattern here, is that you're going to have this one, and this one, which is yellow and wrinkled, which is 3. And lastly, your last one is going to be 2 recessive ones, which is yellow and smooth. So this one is where you actually have just that one left over. And that's usually the pattern that you'll get for heterozygous cross in a dihybrid. So your phenotypic ratio is 1, 3, 3, 1. I'm just have to write it vertically, but that's your phenotypic ratio, 1, 3, 3, 1 with these phenotypes. And if you look at the genotypic ratio, now genotypic ratio is a little bit more complicated, or not more complicated, but more to write, because now you have to look at each individual genotypes. So if you were to look at these genotypes, you would have to separate that this is homozygous dominant, so GG, then you would have to count how many you have. So this is 1, and then that's it, 1, and then you have to count uppercase, and then look, and a heterozygous for this one. So this is 1 here, and then 1 here, so that's 2. And then you can see that genotypic ratio, it gets a little bit more work because you have to really count them all. So then that's your dihybrid cross question. And if you were to finish the genotypic ratio, you're going to get the same thing, except that you're going to have a lot more numbers because you have more combinations of genotype compared to phenotype. So hopefully this video has helped you with your dihybrid cross. Make sure you can relate this dihybrid cross to your law of independent assortment, because that's really where it comes from, and also to your law of segregation for your monohybrid crosses. Thank you for watching. See you later.